Recording in progress.
So good morning, students. Today I'm going to talk about the cell-cell communications. Right. Have you ever imagined how actually the cell in our body, we have so many cells, we have billions of cells in our body, and how these cells actually they like communicate with each other, how they actually provide the instruction to each other, how this protein they actually give the signal to each other. Have you ever wondered how these processes they are running concurrently in our body? Have you ever wondered before?
Yes, hormones.
Yes. Right. Okay, if you look at this, this is just a very small part of the cell-cell communication network. Have you ever seen this before?
No.
No.
Unfortunately, yes.
Oh, yes, that's good. Students, I hope, because I keep admitting people, it's very, very annoying. I hope all of you can attend the class on time, because when I keep admitting people, actually is actually disturbing the way I talk, because I keep admitting people to the group, and I don't know why they cannot come in on time. So I hope students, please cooperate with me. Yeah, come before the class started, and so that I don't have to admit people every single moment. All right. So when you look at this, like I believe Abraham probably have seen this. This is just a very small part of the network. What we call the cell signaling. So if you look at this, remember that this is the membrane bilayer. Can you see this? Can you see my cursor?
Yes.
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 Okay. So this is the membrane bilayer, and on the membrane bilayer, or in between the membrane bilayer, there are so many receptors embedded, whether in or on the membrane bilayer. Yeah. Remember that? Have you seen this before?
Yes.
Yes.
Yeah. So yesterday we talked about the physical environment, right? Like pH, and also the osmolarity. Remember that?
Yes. Yes.
Yeah. So when the pH change, when the osmolarity change, so the receptor or the protein that actually residing on this membrane bilayer will collapse. And that's why they cannot carry out the physiological processes, which is the down regulations here, the downstream signal here. When this, for example, this is the one of the proteins, right? And they are coordinate so many processes downstream, correct?
Yes.
Yeah. So when this transmembrane proteins would, when the functions no longer, the function is disrupted. So all the processes downstream of here will be disrupted as well. So I try to relate it to with the cells, cell culture. So when you look at the cell signaling, this is how the cell they communicated with each other. It's very complicated, right?
Yeah.
Yeah. So if I ask you to draw this diagram and then explain in detail, can you do that?
No.
Probably
Probably
not.
not.
Yeah. This is how I learned in UK before. We have to understand. Of course, my lecturer will never say you have to remember, you have to memorize this. But we know from between the line or the clue or from his mouth or her mouth, we know she or he will expect us to understand this comprehensively. So I ever drew all of this, I remember everything when I did my PhD before, and so we want to understand how actually this. I like signaling pathway because I wanted to know how the proteins. This name, these are all the proteins like Merck, the Riam, the Merck here, the RAS, the Commons, the TARC, or PKC. These are all the proteins. in our body. And each of these proteins, they have their own function. Be it transcription factor, be it adaptive proteins, all of these, they play a very important role. They could, this protein, they could promote, they could also inhibit the expressions of other protein. This is very interesting, actually, if you really wanted to study the biology in the field of cell-cell communications. Okay, so this is just an overview. And for example, for now, we look at from a single cell to a complex structure. People asked me yesterday, some students asked me about genetic mosaic, right? They wrote to me. I can't remember who the person is. Yeah. They asked me, but that one I will explain later. Let's look at this from a single cell to a complex structure. So while each organism started off as a single cell, all of us start from a single cell, right?
Yes.
Yes.
Yeah, it's from the fusions of the nucleus from the sperm and also the egg, and then become the zygote, and then that's from the single cell. And the progeny of that cell, you know the progeny? Maksud progeny tahu tak?
Offspring.
The offspring, that's correct. The offspring of that cell form complex structure tissue and organ that are themselves integrated into the larger system. That's why from a single cell, from the zygote, then slowly become embryos, and slowly becomes a complete organism. But just imagine, you just ask yourself, when the egg and sperm form under the single cell, there are no structure like hand, mouth, brains, or blood, or other tissue, right?
Yeah.
But
Yes.
where and how this, the complex structures is formed? How did they form? Where they get the instruction from to direct them to become blood, direct them become brain, or other appendages, or bones? Where they get the instruction from?
Gene expression.
Yeah, who instruct them? Yeah, from the gene expression. Who actually instructed them to give signal, to promote?
The brain?
No, not the brain. Single cell mana ada
Cell.
brain? Masa single cell itu? Mana ada brain lagi? Betul tak? Masa the fusions of the nucleus from the
Betul.
egg and the fusion from the sperm kan, dia menjadi satu single cell tu. Satu single cell tu mana ada brain lagi, betul tak?
Oh, okay. Betul.
Ya, tak ada apa-apa form lagi. Tapi semasa dia single cell tu, siapa yang memberikan instruction? Memberikan arahan kepada sel-sel ini untuk menjadi sel-sel yang tertentu.
Protein.
Seronok tak tanya soalan macam ni? That make you think, right? So when you learn developmental biology, there's a lot of questions that you need to ask. And you boleh jadi gila kalau you banyak tanya. And then the more you learn, the more you don't know. Betul tak?
True.
Okay. So there are at least two components to every inductive interaction. So the first component is what we call inducer. Inducer ialah yang sesuatu yang buat induce. Induce dalam BM apa? Yang contohnya yang start something lah. Induce a tissue that produce a signal, or a signal that change the cellular behavior of that of the tissue. So the second components of the tissue being induced is what we call the responder. So something yang induce dia, mesti ada something yang respond kepada dia, betul tak?
Betul.
Yes.
Okay. So kalau kita tengok kan, kalau kita tengok picture ini ialah head and this is the trunk. So this is the normal inductions of the lens by the optic vesicle. Yang ini kawasan di mana dia menghasilkan optic vesicle, ia membentukkan mata kita. Okay, dan sekiranya optic vesicle cannot induce ectoderm that is not component. Contohnya kita ambil ini, kita keluarkan, kita excise yang ini, kita potong bahagian sini dan kita letak di sini. Boleh tak pembentukan lens kanta mata kita dekat sini? Boleh tak?
Tidak.
Tidak.
Tak
Kerana
boleh.
dia tidak ada responder, betul tak?
Betul.
Okay. Dan sekiranya the optic vesicle is removed, okay, is removed, no lens is, kalau kita potongkan dia, jadi lens pun tak terbentuk, betul tak?
Betul.
Ya, kerana tidak ada signal daripada optic vesicle di sini. Dia tidak memberikan inducer, dia tak induce. Jadi kalau dia tidak ada yang memberi induce, maka pun tidak ada yang akan terbentuk. Betul tak?
Betul.
Okay, so tissue other than optic vesicle is implanted, no in the. Contohnya kalau kita ambil tisu yang lain, kita letak di sini, pun tidak ada kanta yang terbentuk, betul tak? Tak ada induction yang berlaku, betul tak?
Betul.
Okay, so apa yang boleh kita deduce, boleh kita rumuskan daripada picture ini ialah sekiranya sesuatu itu yang memberikan induce yang start something, dia kena ada responder dia. Okay, dia mesti like this is how they communicate lah. Kalau you cakap dengan dinding tidak akan respond dengan you tak, because dia tak induce. You induce dia tapi dia tak respond, dia tak faham. Tapi kalau you cakap dengan orang lain, dan dia faham. You tahu tak analogi ini? Boleh faham analogi ini?
Boleh.
Faham. Boleh.
Chapters and understanding.
Yeah, so this one kan kita kena faham ya. So banyak eksperimen boleh dimanipulate. Alright, kita boleh banyak manipulate. This is how we did before lah. Saya pun pernah buat di sini. Kita dapati bahawa dia kena ngam, orang kata. Dia kena secocok, orang kata. Kalau sel yang berdekatan itu dia tidak faham, dia tidak boleh respon, tidak boleh communicate, maka dia tidak akan membentuk sel yang perlu dihasilkan itu. Ya. So sel-sel yang berdekatan biasanya dia boleh communicate with each other. Alright?
Okay.
Okay.
So, this ability to respond to a specific inductive signal is called competence. Maksud dia mampu, dia competence. Okay, dia boleh respond to each other. Jadi, ini kita katakan dia competence ya. So, competence is not a passive state, but an actively acquired condition. Dia memerlukan keadaan macam itu untuk pembentukan struktur-struktur badan yang seterusnya. Contohnya kita ambil satu contoh ya. In the developing chick and mammalian eye, the PAC6 protein, ya, PAC6 ialah sejenis protein morphogen yang mana dia appear to be important in making the ectoderm competent to respond to the inductive tissue from the optic vesicle. So ini structure A di sini ialah wild type. Wild type maksudnya yang normal lah, ya, yang normal. Yang ini ini contohnya ini ialah mouse lah, mouse, the baby mouse, the embryos. You can see kalau kita buat sectioning, limit brain dia okay, forebrain land pits kita nampak land pits dia, okay, dan lateral nasal prominence, the nasal pate, and the medial nasal prominence, kita nampak dia actually dia all intact, dia lengkap ya kalau kita buat sectioning. Ya, kita buat sectioning macam ni. Right? Boleh?
Ya.
Kalau kita tengok B dengan D ini ialah mutants, PAC6 mutant. Yang mana baby dalam embrio ini dia tidak ada protein PEG6. Maksudnya PEG6 dalam embrio di sini untuk Kennedy ini kita sudah knock down dia. Dan apa yang berlaku kita nampak di sini bahawa tak nampak lipids, betul tak?
Betul.
Yeah.
It doesn't have eyes.
Yeah, doesn't have eyes. So apa maksud di sini? Apa fungsi PEG6 ini in the development?
Inducer.
PEG6 play very important role in the developing of eyes, betul tak?
Yes.
Betul.
Inilah bagaimana kita nak decipher, kita nak mengetahui fungsi sesuatu protein ataupun gene. How do we decipher the function of the gene? Ialah dengan menggunakan kita knock down dia. Kita nak tengok apakah fungsi yang dimainkan ataupun peranan yang dimainkan oleh PEG6 protein ini. Boleh faham kan?
Boleh.
So bila lain kali kalau ada yang kamu berhasrat nak menjadi developmental biologist nanti ke ataupun geneticist ke, inilah something yang very interesting yang boleh kamu faham. Kerana kita masih jauh lagi nak memahami fungsi-fungsi untuk semua protein ini. At the end, all the functions of the gene will be deciphered. We are towards that direction. So nanti kalau sudah siap semua tu, then the gene network yang menyeluruh akan dihasilkan dan kita akan nampak the gene circuit, the complete gene circuit nanti. Okay? Dan kalau kita tengok from a single cell to the complex structure, so this ability to ini kita tak nak baca ini sama lah macam yang apa tu slide yang lepas. Tapi kalau kita tengok di sini wild type, okay? Kalau kita tengok wild type di sini, this is the optic vesicle, surface ectoderm and lens induction. Kita nampak wild type, kita nampak okay kan, semua okay. Okay?
Okay.
Okay.
Tapi kalau kita tengok optic vesicle untuk PEG6 negatif ni, apa yang berlaku? So, okay, surface ectoderm dia nampak okay, and then the lens induction nampak okay. Tapi kalau kita tengok PEG6 negatif maksudnya tak ada PEG6 di sini dan surface ectoderm PEG6 tak ada di sini, maka tidak ada lens induction.
PEG6 has to be present in the surface ectoderm for any lens.
For the
to form.
inductions of lens, that's correct. Faham tak? Kerana walaupun PEG6 dekat optic vesicle ni tidak ada, tetapi dia wujudkan pada surface ectoderm, dan lens induction masih berlaku lagi kan?
Yes.
Yes? Is that correct?
Yes. Correct.
Yeah.
So what does it mean is the PEG6 must be present in the surface ectoderm to allow the lens induction. Faham tak? You deduce the maklumat daripada foto macam ni.
Faham.
A lot of people they don't understand how to decipher the information from the picture that we show to them. Because kalau kita tengok kan, as long as surface ectoderm tu dia ada PEG6, ya, dia ada PEG6, maka lens induction ni tetap berlaku lagi kan? Walaupun PEG6 missing pada optic vesicle. Correct? Kalau kita tengok dua ni. Dan kalau kita tengok di sini ya, kalau surface ectoderm dia tidak ada PEG6 protein, walaupun dia ada PEG6 ni ada pada optic vesicle, dia tetap tidak ada lens induction, betul tak?
Yeah.
Ya. Correct?
Yes.
So, apa conclusion kita ialah?
PEG6 needs to be present. on the
Surface ectoderm needs PEG6.
For?
lens induction.
Yes, that's correct. Macam itulah kita decipher. Kita mendapat maklumat. The picture mean nothing if you don't know how to decipher the information from it. How you don't know how to extract the information from it. Correct?
Correct.
So kita kena faham, kena bijak, kerana contohnya this picture will be meaningless kalau kita tak tahu bagaimana kita nak compare dia, betul tak?
Betul.
Ya. So kalau kita tengok di sini, kita letak dia macam ini dan kita susun, kita faham dengan jelas. Ya, kita faham dengan jelas bahawa dari sini terus kita nampak bahawa bila PEG6 tidak ada pada surface extodome, memang lens induction tidak berlaku. Betul tak?
Betul.
Betul.
Ya. So boleh faham? Saya gunakan bahasa yang layman supaya kamu orang ni boleh faham dengan senang. Kerana this subject honestly dia sangat dry, betul tak?
Yes.
Ya, kalau saya baca saja you all tidak akan faham, betul. So saya lebih suka terangkan subjek ini dengan guna bahasa yang senang kita faham. Dengan contoh-contoh yang lebih senang faham supaya you all senang faham juga. So kalau ada yang tak faham, then nanti you bagi tahu lah. Ya, kita boleh discuss further. But so far okay tak?
Okay.
Okay, doktor.
Okay, ha? Okay, good. Now, kalau kita tengok, apa itu reciprocal, apa itu sequential induction? Ada dua jenis untuk induction. So, another feature of induction is the reciprocal nature of many inductive interactions. So, once the lens has formed, it can then induce other tissue. Faham ni? Faham this statement? So, one of these responding tissue is the optic vesicle itself. So, under the influence of factors secreted by the lens, the optic vesicle becomes the optic cup, and the wall of the optic cup differentiates into the pigmented retinal, then the neuroretinal. So, apa yang dimaksudkan ini ialah sequential ialah A induce B, B induce C. Boleh faham?
Boleh.
Kalau reciprocal, apa maksud reciprocal itu? Pernah dengar perkataan reciprocal?
Responding to. Responding to.
Betulkah, Abraham? You sure?
Up and down?
Huh?
Corresponding.
Apa maksud reciprocal?
A response.
You baca betul-betul.
Respon. Mutual action.
Okay. Kalau kita tengok di sini, cuba tengok. So under the influence of this lens, the corneal ectoderm cell become columnar and secrete the multiple layer of collagens. So in the mesenchymal cells from the neural crest use this collagen matrix to enter into the area and secrete the set of proteins, which include the enzyme of hyaluronidase that's further differentiate into the cornea. So the third signal, the hormone tyrosine dehydrates the tissue and makes transparent. So kalau kita tengok di sini, the prospective ectoderm, dia akan menjadi prospective neuroplate. Okay? And then dia dengan adanya dengan ini late gastrula and early neurulation, and kita tengok stage di sini lah ya, and then dia akan menjadi prospective optic cup, optic vesicle, and then optic cup terusnya, dan pigmented retinal. So ini kita katakan dia ialah sequential. Dia dalam satu sekuen yang berterusan, betul tak?
Betul.
Step by step.
Ya, dia dalam satu siri jujukan yang berterusan kan. Dia step by step kan. Betul?
Yes.
Kalau reciprocal, dia lain. Dia bukan step by step, tapi dia ada cabang-cabang dia. Ya, contohnya di sini prospektif pharynx masuk, prospektif heart masuk, baru dia di sini prospektif epidermal akan menjadi sini. And then the prospective neuroplate akan induce the prospective epidermis menjadi prospective lens in ectoderm. Ini dikatakan reciprocal. Dia lain dengan sequential. Nampak tak?
So reciprocal means that it needs extra components, or
It is not in the sequence. It's not in the step-by-step punya process.
happens randomly?
It's not like happen randomly, but dia macam like A induce B, B induce C, and C can induce A.
In a circle?
It's not also in a circle. Dia bukan step by step lah maksud dia. Okay, you see? Okay, dengar. A induce B, B induce C. C sepatutnya induce Z, betul tak? Kalau sequential.
Yes.
So,
Yes.
kalau A induce B, B induce C, C induce A, and A induce D, induce reciprocal. Dia bukan dalam satu jujukan ataupun step by step punya cara.
It's like that Krebs cycle where you have stuff coming in and coming out.
Ya. Macam yang saya show dengan kamu inilah. Ya.
Basically,
Dia
it's
bukan-
complicated.
Yeah, it's a bit complicated than the sequential punya induction lah. Boleh?
Okay, boleh.
Dia memang lain dengan sequential. Alright? So, signaling system. So, inducing factors are commonly proteins. Okay, kita memang proteins lah, because gene make proteins kan. Although a few are small lipid soluble molecule. So, most are secreted by the signaling cells, although some are integral membranes protein that can only interact with the immediately adjacent cell. Adjacent cell ialah sel yang berdekatan lah. So, they bind to a specific receptor and it activate the signal transduction pathway within the cell. Within the cell yang di dalam sel itu sendirilah. So, this may lead to the activations or progressions of the specific genes or to change in cell behavior mediated through the cytoskeletons or through the change of the cellular metabolism. So, ingat signaling system ini dia sangat complicated. Dia bukan sesuatu yang guna satu ayat boleh describe everything. Okay, nanti saya akan show kamu contoh-contoh lah. Tapi before that, cuba kita tengok apa tu paracrine factor. So, how are the signal between the inducer and responder transmitted? Bagaimana the signal? Signal di antara inducer dengan responder ni dia boleh transmit di antara satu sama lain. And many of these paracrine factors can be grouped into the 4 major families on the basis of their structure, and these family are like the fibroblast growth factor or the FGF family, the hedgehog family, the wingless WNT family, and the TGF-beta superfamily. Okay, so ini antara 4 family yang memainkan peranan yang sangat penting lah dalam development. So, kalau kita tengok, okay, so the paracrine factor ialah diffusions or inductions on one cell to another. Yang ini kita kenali sebagai paracrine factors. Kalau matrix, dia keluarkan matrix 1, the cell induced chain in another cell, and contact between the inducer and the responding cell, dia dah berbeza. Ini tengok cara yang berbeza di bagaimana sel itu communicate. Ya, paracrine factor merupakan salah satu daripadanya. Lepas tu pengeluaran matrix, penghasilan matrix satu, and then satu lagi di mana contact between the responder and the inducer. Boleh?
Boleh.
Boleh.
Okay. Nah, contohnya the paracrine factors di sini. This is a cell membrane. This is TGF beta family. We have the type 1R and type 2R. R is a receptor, and this, for example, the smart proteins. they bind to the type 1R, and then they become phosphorylate, and phosphorylate, then they go for the genes regulations. FGF pun sama. Okay, and sorry. Okay, the insulin IGF family pathway pun sama, and then the Hashop pathway. Alright, ini merupakan satu simplify punya figure or illustration for you to look at the four super family yang very important in the cell communication. Do you know how to read the signal? For example, macam ini, kita ada anak panah yang terus kan?
Yes.
Ya,
Okay.
this yang ini ialah dia berinteraksi terus. Okay? But yang kalau macam ini, nampak tak dia ada macam satu, how to say this yang macam
Dead
ini.
end.
Dead end ini, this one is like dia inhibit or suppress. Like PKB suppress the FOXO, the HS suppress the PTC, and then the PTC suppress the the SMO. Dia suppress. Kalau kita tengok signal macam ni, palang macam ni. Okay? So you kena faham bagaimana sel ini dia berinteraksi. Sama ada dia promote ataupun dia inhibit. Ini yang kita kena faham. Kamu kena faham bagaimana nak baca dia lah. Boleh ya?
Okay.
Boleh.
Okay. Ini pun sama. Ya, the canonical wing pathway, the notch pathway, the inflammatory system, another wing pathway, and cytokines system. So the transforming growth factor superfamily, kita banyak. Okay, the fibroblast factor, and then the Hashop family, the wing family, the cell, okay, the RTK pathway, smart pathway, and Jackson pathway, and then the Hashop pathway, okay, cell death pathway, apoptosis, okay, and notch signaling, okay. These are the signaling system yang ada yang saya perkenalkan dengan you all, but I will not go through one by one because you will do the assignment. Okay, you'll be doing the assignment. Then let me show you. Hang on.